Hello viewers, welcome to this particular lecture on Introduction to Photosynthesis. In this particular lecture, I shall be covering different aspects of photosynthesis. I shall be talking about the definition of photosynthesis, followed by the site, that is the organelle involved in photosynthesis. I shall also be talking about the different pigments that play important role in photosynthesis, and then I shall be briefly discussing the different phases of photosynthesis, that is the light phase and the dark phase. And towards the end, I shall be talking about the different important aspects of photosynthesis. So as the name signifies, photo meaning light and synthesis meaning preparation or formation of something. Photosynthesis is a process which actually involves synthesis of sugar in presence of sunlight in the green chlorophyllous tissue of plant. Actually, there are two phases, the light phase where the light is helping in the synthesis of different energy related compounds, namely ATP and NADPH2, and that energy is utilized during dark phase for carbon fixation and production of sugar. The process of photosynthesis is actually a photochemical process taking place in the green chlorophyllous tissue of plant in presence of sunlight, where they procure light and of course carbon dioxide and water from nature, and they produce sugar or hexose sugar and produce oxygen as a byproduct. Of course, there may be another way of defining photosynthesis. We can say that the kinetic energy of the photon particle is being transformed to potential energy in the form of energy rich compounds, and finally it is stored as chemical energy of sugar in the green plant. Of course, it can also be said like this: it is the conversion of inorganic carbon of nature, that is carbon dioxide, to organic carbon, that is sugar, in the plant tissue. Actually, photosynthesis can be of two types, which is on the basis of release of oxygen. Accordingly, the process which releases oxygen is called oxygenic photosynthesis, and if it is not released, it is called anoxygenic photosynthesis. In oxygenic photosynthesis, actually the process is involving the green chlorophyllous tissue of plant. It is also taking place in the cyanobacteria, where clearly there are two photosystems, namely the P680 and the P700, and the P680 is involved in the breaking of water molecule and release of oxygen. And the electron liberated from P680 through various carriers come to P700, whereas the electron liberated from P700 is coming to NADP and is reducing that. So in the process we get oxygen and also we get water. But in case of anoxygenic photosynthesis, which actually takes place in some other bacteria, of course photosynthetic bacteria, that particular oxygen is not released. And it is shown by different bacteria like purple bacteria. And in this purple bacteria, what happens is there is a photosystem which is called P870, and it liberates the electron and it is coming via different carrier. Finally, it is received by NADP. At the same time, the electron that is replenished in P870 comes from H2S or iron compounds. So as a result of which, what happens is, in the process, sulfur or iron may be liberated. In another bacteria, namely Chlorobium, there is a unique thing observed. What is that? The P840 is the photosystem which receives the light. It liberates the electron, and this electron via FES and cytochrome B comes back to P840, but it is not directly coming back in the way it is received by NAD. So the electron replenishment here also comes from H2S or S2O3, and thereby here also oxygen is not liberated. Instead of that, sulfur is liberated. So we can always compare that photosynthesis, which is liberating oxygen, oxygenic, and anoxygenic, has some definite points of difference. It includes the photosynthetic pigment. Of course, the photosynthetic pigment in oxygenic photosynthesis will be chlorophyll, the different chlorophyll A, B, etc. And for that of the bacterial and oxygenic photosynthesis, it should be bacterial chlorophyll. Of course, in this case, that is in oxygenic, there will be two distinct photosystems, PS1 and PS2, but in case of an oxygenic photosynthesis, there is only one photosystem and the cyclic photophosphorylation is only operating. Here the electron donor in case of oxygenic photosynthesis will be water, but for an oxygenic process in case of oxygen or other compounds. Of course, it will be producing oxygen and it will be producing sulfur in case of anoxygenic photosynthesis, and the primary energy converting processes will involve the production of ATP and NADPH for oxygenic one, and for anoxygenic one only ATP will be generated. Photosynthetic organelle, that is the plastid. Actually, plastid remain as proplastid, which is a non-membranous structure which gradually develop into different forms of plastid. And this include the chromoplast, the leucoplast and the chloroplast. Actually, the proplast, if it receives adequate sunlight, it will be converted to chloroplast, it will have chlorophyll. But if it is not receiving adequate sunlight, it will be converted to leucoplast and it will act as a storage organ. On the basis of the nature of the storage compound, it can be amyloplast, which is storing starch. It can also be elaioplast, which is storing lipid, and it can be proteinoplast or aleuronoplast, storing the protein aleuron grains. Similarly, chromoplast can be orange or yellow in color because it is having pigments of xanthophyll and carotene. Of course, the green chloroplastid is the most important organelle. This organelle is the site of light phase, that is in the grana, and the dark phase occurs in the stroma. But this particular organelle will have unique granule and intergranule region, and it will have DNA, it will also have starch granule and stromules. What happens is, with the gradual degradation of the leaf, when the plant gets old, then this particular chloroplastid is actually getting degraded, and this is degraded into red colored structure. What happens is, there will be appearance of some membranous sac, there will be appearance of carotenoid, there will be disappearance of chlorophyll, and gradually there will be stromules appearing, and the content will be getting decomposed and the plant leaf ultimately will fall off. There is a unique process by which these particular plastids can be converted to other forms. Like, say for example, if it is a particular chloroplastid, the chloroplastid may be converted to senescent chloroplast, which I have already stated, but a chloroplast can be formed from chromoplastid, or a chromoplast can be formed from amoeboid plastid, which is unique. What does it mean? It means that the proplastid, through stages of conversion and interconversion, ultimately produces chloroplastid, which is the ultimate goal, and in between they will produce different other organelle, which is either chromoplastid or different forms of leucoplastid. But one thing is definite, that this particular chloroplastid will always have this grana or membranous region within, which will definitely increase the membranous area and photosynthetic light absorbing service. But the chromoplastid, that way they do have pigment, but they do not have internal membranous structure. At the same time, the chloroplastid itself can be of different types. There may be the chloroplast, that is the compound chloroplast, which is present in different forms of green algae. Like in Chlamydomonas, the chloroplast is cup-shaped. In case of Spirogyra, which is a filamentous algae, the chloroplast is spiral, or it can be reticulate in case of another filamentous green algae, which is Oedogonium. So this particular chloroplast structure is a compound one, and it will have multiple starch grain and proteins stored in it, which are called pyrenoids or protopyrenoids. And depending upon the nature and structure and shape of the chloroplast, the algae can be easily identified. But if we come to chloroplastid structure itself, it is highly developed, it is unique discoid structure having a double membrane, and this double membrane structure is having an intermembranal space within. Inside the chloroplastid, there will be stacks of thylakoid producing the grana, and the grana are interconnected by lamellae, which is also called the stroma lamellae. What happens is, actually the chlorophyll molecules are arranged inside this particular grana in the form of quantosome, and this quantosome actually is formed of say 250 chlorophyll molecules or more than that. But what is important, there is a unique structure resembling bacteria, and that includes the 70S ribosome and the DNA. The DNA within the chloroplastid and the presence of 70S ribosome, they do indicate that this plastid is not produced from the cell itself, but it has been acquired by some external bacteria, maybe because of the process of endosymbiosis. Now, when I say bacterial pigments, of course it is bacteriochlorophyll C, bacteriochlorophyll D, E and G. Mostly it is C, D, E present in different green bacteria, but bacteriochlorophyll G is present in heliobacteria, which make them survive in high intensity of light. But when you talk about bacteriochlorophyll A and B, bacteriochlorophyll A is present in purple bacteria, but bacteriochlorophyll B is present in Rhodopseudomonas and other forms of green bacteria. But when we come to higher plant, the chlorophyll A, B, C, D and E. Chlorophyll A is known as the universal photosynthetic pigment because it is present in all plants and algae, that is green algae or other forms of algae, but not in case of bacteria. Chlorophyll B is present only in higher plant and green algae. Chlorophyll C is present in diatoms, dinoflagellates and brown algae, whereas chlorophyll D is present in red algae and chlorophyll E is present in yellow-green algae or in the zoospores of Vaucheria. There is a basic difference between the chlorophyll formula. Now, as I told you, the basic chlorophyll molecule, which is the universal chlorophyll molecule, is chlorophyll A, and it has a formula of C55H72O5N4Mg. Chlorophyll B, which is producing starch, is having a formula of C55H70O6N4Mg, and there may be chlorophyll D or E, which is almost having the same type of formulae, but chlorophyll C1 and C2 is having less carbon. Here the chlorophyll C1 formula is C35H30O5N4Mg, and chlorophyll C2 is having a formula of C35H28O5N4Mg, which indicate that chlorophyll C is definitely different from the other forms of major chlorophyll like A, B and D. The chlorophyll molecule actually is having two different parts, the head and the tail. The head is formed of magnesium, porphyrin, hydrophilic head, and the tail is formed of lipophilic phytol tail. Actually, the head has magnesium at the center, and there are 4 pyrrole rings attached to the magnesium by nitrogen. And to the 4th pyrrole ring, the phytol tail, that is the hydrocarbon tail, is attached. Basically, the 2nd pyrrole ring is important because to this particular pyrrole ring, there is the R functional group for chlorophyll A, which is the universal photosynthetic pigment. The R stands for CH3 or methyl group, but that particular chlorophyll B will have a R of CHO, aldehyde group. So, there is a basic difference between chlorophyll A and B in their structure, though the broad structure remains the same, that is the hydrophilic porphyrin head and the lipophilic phytol tail. Chlorophyll biosynthesis starts from glutamic acid with the functional glutamyl tRNA, and it is converted to aminolevulinic acid, and then with the dehydration or dehydratase enzyme, it is producing porphobilinogen, and then it is producing hydroxymethylbilane, and then uroporphyrinogen, coproporphyrinogen and protoporphyrinogen. At this point, the protoporphyrin 9 is produced, which ultimately will be taking up magnesium and iron to produce magnesium protoporphyrin, which ultimately will produce chlorophyll, and protoporphyrin 9, if it takes up iron, it will be producing phytochromobilin. Let us talk about the different types of carotenoids. Carotenoids can be lycopene, which is red in color, alpha carotene, which is yellow in color, beta carotene, which is orange, and gamma carotene, which is red-orange in color. And carotenoid also have a long chain with two hexagonal ring at the two terminal ends, and more or less the pattern is same for alpha and beta carotene. Only for gamma carotene, one of the chain is open. The carotenoid biosynthesis starts from glyceraldehyde 3-phosphate, which is producing deoxyxylulose 5-phosphate, and then gradually it will produce dimethyl allyl diphosphate, geranyl diphosphate, phytene, and then eta carotene. Eta carotene undergo desaturation to produce lycopene, and then this lycopene will be producing two types of pigment. On one side, it will be producing the beta cyclase action and it will produce the lutein, and on the other side it will also undergo cyclization to produce beta carotene, zeaxanthin, and in this way we can see that carotene will be converted to xanthophyll, which is yellow in color. Now let us talk about different types of xanthophylls. Actually, xanthophylls include the lutein, which is yellow in color. Apart from that, there is violoxanthin, which is yellow in color, beta cryptoxanthin, which is orange in color, zeaxanthin, which is yellow-orange in color, and astaxanthin, which is red in color. In fact, these particular pigments are having more or less the same pattern of red or reddish-yellow in color. Now, the xanthophyll interconversion is very interesting. It starts from phytoin, and this phytoin will be converted to eta carotene, and eta carotene will produce the red pigment of lycopene, which is present in tomato, as you know. Under the action of LOR1 gene, it will be converted to beta carotene and alpha carotene on each side, and then beta carotene may produce the zeaxanthin, anthraxanthin, violoxanthin, neoxanthin. These are all yellow in color. And on the other hand, alpha carotene might produce lutein and loroxanthin, which is reddish yellow in color. Phycobilins are so called because they are bile-like pigments present in algae, and they include the phycocyanin and phycoerythrin. The phycocyanin and phycoerythrin, they are more or less the same. They are the straight chain compound having 4 pyrrole ring, but only there is a difference in their side chain, that is the last terminal group or last terminal functional group for phycoerythrin is CH2CH or CH-CH2, and for phycobilin or phycocyanin it is CH2CH3. Now let us talk about the energy transduction during photosynthesis. Actually, the photosystem 2, which is having the chlorophyll B or P450, is giving the electron to lutein or P470, then zeaxanthin P480, beta carotene P500, then lycopene P510, and then chlorophyll B, which is P650, and finally it comes to chlorophyll A, which is P680. So electron is gradually being transferred from the second photosystem to the first photosystem, but in the process they lose energy following the second law of thermodynamics. But there is adequate energy present to liberate the electron from the PS1, and thereby there will be photolysis of water or production of reduced NADP. So this is how we can see that energy which is not coming directly to chlorophyll, they are coming to other pigments, and these pigments are ultimately transferring it to chlorophyll, and thereby the photosystem is activated, and this phenomenon is called antenna pigment absorption. We can see that the photo reactivation center is just like a funnel having different other carriers in the outside rim and the chlorophyll remain at the center, which will gain all the energy. So in fact, the energy which is absorbed by pheophytin, plastoquinone, cytochrome, they are all ultimately coming to chlorophyll, and thereby the chlorophyll is getting activated, and that will give rise to the electron, and that ultimately switch on the cyclic photophosphorylation. Photosynthesis is having two definite phases, the light phase and the dark phase. The light phase operate in the membrane of the thylakoid, where there will be the two photosystem, the PS2 and the PS1. The PS2 will give rise to the electron, which will come to PS1, and the PS1 will give rise to electron, which will be ultimately accepted by NADP. But water, which is actually undergoing photolysis, will give rise to H plus ion, and oxygen will be liberated out. But this H plus ion ultimately will be coming out via specific ATP synthesis machinery, which is called ATP synthase or ATP synthetase, and thereby the ATP is produced. So we can see in the light phase there are three major end product, which is oxygen, which is ATP, and reduced NADP. The last two, namely the ATP and the reduced NADP, will be utilized during dark phase, and the dark phase actually is operating in the stroma, which is having definite processes like carbon fixation, reduction, then synthesis of hexose, and regeneration of the starting compound, which is RUDP. This entire process is operating actually with the help of energy generated from ATP and reduced NADP. Photosynthesis has got some immense future. Actually, the production of hydrogen, which is the future fuel, or methane, may be the product of photosynthesis. And already there is enough research carried out in Finland, where they are trying to utilize green algae photosynthesis to liberate hydrogen, and that will produce the future fuel. Apart from that, photosynthesis is also important in different other ways, which include it is the source of food, it is giving energy in the food chain, it is maintaining the balance between oxygen and carbon dioxide. At the same time, it is having enough oxygen to allow the other life forms to survive. Photosynthesis will also give rise to different plant byproducts which are utilized by mankind. And of course, the fossil fuel, which is the photosynthesis activity of the yesteryear plant, is being utilized now by human being in the form of natural gas, coal, and petroleum. So what we can see that photosynthesis is immensely important in the present day life, and without photosynthesis there will be no life in this earth in the present day. So what we have discussed actually is the basic photosynthetic process, definition of photosynthesis. We have also understood the different sites or the chloroplastid and their evolution in photosynthesis. We have talked about the different chlorophyll pigments and their pattern or their biosynthesis, that is how they are interconverted during the biosynthetic process. I have just given an overview of the actual photosynthetic process, and towards the end I have also talked about the importance of photosynthesis. Thank you.